So the title of my talk that I've been invited to present is Next Generation Tools of Bioinformatics. And there's a particular reason why I've included Next Generation in the title. And that's because a lot of the tools that I'll be discussing throughout this seminar actually have really been made possible only through the generation of large amounts of human sequence data. And so by accumulating this sequence data from the human population, we are now able to begin understanding what are the normal patterns of genetic variation in samplings of the human population. And by knowing what are these typical patterns, it enables us to also identify what are atypical. So when we see some genotype that's exceptional, that's outside of the range of what we usually expect for a given gene. And I'll describe a few of these bioinformatics signatures throughout the talk in certain contexts. So the talk will be broken up into three sections. First one, I'll be discussing the N of 1 paradigm. Many of you from clinical departments would be familiar with this. It's essentially when you look for, within a given trio, where the goal is to try and understand what's happening in that one patient and try to understand what the underlying genetic cause is for their ascertained phenotype. Then I'll be discussing a case control paradigm, which really uses the power of numbers when you've accumulated enough of this case population and you have sufficient power to try and ask whether you can, with statistical rigor, identify new and securely implicate new disease genes in human genetics. And finally, I just want to spend a bit of time introducing you to some of the work that we've done of using our ability to understand the patterns of genetic variation in non-coding sequence to help us better interpret variation that happens outside of the human protein coding sequence. So in terms of the N of 1 setting, many of you will be familiar with this. This is, in general, you take parents, unaffected parents, who have a child who has sporadic onset of generally a severe disorder, and you exome sequence the trio to try and understand what's happening in the child. When you do this, you end up with a long list of variation, genetic variation. It's in the hundreds of thousands of variants in our protein coding exomes. But what we do is then we try to identify qualifying genotypes. And I want to pause and emphasize a bit on what I mean by qualifying genotypes, because this will be a term that I'll use repeatedly throughout this seminar in a number of scenarios. Essentially what qualifying genotypes are is a way that we impose certain criterion on the list of candidate variants to subset it to a subset of variants where we believe that we are enriching for real risk alleles. And we can use certain information to filter that. The two most fundamentally important ones being population frequency of the variant or genotype in the population, and also what the predicted effect of the variant is. Is it non-synonymous, synonymous, protein coding, non-coding, etc. When we do this in the setting of trios that have been ascertained for undiagnosed genetic disorders, on average, we identify about one germline de novo mutation in a trio. And so these are mutations that have arisen de novo in the germ cells of the parents generally. So parents are non-carriers for these variants. On average, we identify one hemizygous variant in male probands, about one homozygous qualifying genotype, and about two compound heterozygous genotypes. And I have to stress that this is using the conservative filters that you would generally impose when you're looking at these very severe early onset infantile genetic disorders. The combined set of 4.5 can vary quite a bit, and I'll discuss some reasons why, but one of the main reasons is it can vary a lot based on the genetic background of that patient's family, and we'll discuss that later. But essentially you end up with half a dozen to a dozen qualifying genotypes per trio, and sometimes one of those genotypes will be a genetic diagnosis, but most of the times they end up being genetic candidates, variants of unknown significance, and I'll describe both of these classes. So to make it as a genetic diagnosis, there's actually quite a bit that it has to, criteria it has to fulfill. And I've highlighted two references that I strongly encourage anybody that's interested in this to follow up and read, and I'll pass it on to Alana afterwards to distribute around the two references. But in a nutshell, the way it works is, firstly, the mutation that you see in the patient has to occur in an established human disease-associated gene. Moreover, the phenotype of the patient has to be similar, judged to be similar with previous reports of what mutations in this gene have resulted in. Moreover, the genetic model has to be consistent, so you would expect to identify recessive genotype if that gene's only ever been linked to human disease through a recessive inheritance model. Moreover, the qualifying mutation itself has to have been observed in other patients or the same class of mutations have to be observed. So, for example, if you identify a mid-protein loss of function variant in a human haploinsufficient disease gene, then that can often be sufficient, but you would want to do the sanity check of ensuring that that actual loss of function variant results in loss of function through some functional assay. And finally, preliminary, just before final, and probably the most important part is that you would need to have consensus between both the genetics and the clinical team that they both agree that that mutation could be the cause of the patient's phenotype. So the genetics team has to be convinced by the human genetics that's behind that variant, and the clinical team has to be convinced that that variant is consistent with, their patient phenotype is consistent with what's known about human disease from that gene. And finally, often you would want to independently Sanger validate that because Sanger validation is an independent sequencing technology to what's used to identify it when you use whole exome sequencing. And this is particularly important for de novo mutations. When this is done across various groups and publications, the estimate of the diagnostic yield from whole exome sequencing of trios in the context of undiagnosed genetic disorders converges to about 27%. So that's about 27% of these trios are explained by about 1.5% of our genome, the part that 1.5% that actually encodes for the proteins. As I mentioned before, the majority of variants, however, land in the candidate genotype, the genetic candidates, variants of unknown significance, and these are of particular interest for research follow-up. And so, as a community, we've actually gotten fairly good at understanding and predicting, using various in silico measures, which variants are likely to affect the protein, either using protein structure, function, homology, comparative genomics, phylogenetic conservation, etc. And so I've listed 3 of the arguably more popular ones, such as polyphen-2, SIFT, and GERP, but there are many other flavors, and there's a wealth of tools available to try and identify, or to try and determine whether a variant is likely to damage the protein. One distinction that has to be made, however, is that the likelihood of a variant damaging a protein is not synonymous with the likelihood of that variant being pathogenic. And I think this can sometimes be misunderstood in literature. Things like polyphen assess how probabilistically that variant is to damage a protein, not how likely that variant is to be pathogenic or to be damaging. And this is most emphasized by if we look at any one of our exomes, we will find tens of thousands of missense variants that are predicted to be damaging or deleterious or occur in very highly conserved sites in our exomes. And so while sensitivity has been found high for these tools, specificity is quite poor. And so one thing that's been lacking here is an actual appreciation for the genetic background for where these mutations happen. And we have understood as a genetics community that different genes in the human genome tolerate functional variation to different degrees. And so if you see a probably damaging variant in one of the olfactory receptor genes, this variant is probably not consequential, probably not going to result in a clinical phenotype, and will often be allowed to become common in the general population. If you see a probably damaging variant for example, like a calcium channel gene, it's highly unlikely that that's going to become a common variation based on what we know about these genes. And so this kind of motivated us to actually assess this formally genome-wide and to assign each gene, or to assess the relative intolerance of each gene in the human genome. And we did this just by looking at the patterns of standing variation in a subset, or in a sampling of the human population. So we took 6,500 exome sequence samples that were sequenced by the exome sequencing project over at Washington. And from the sequence data, we took just 2 features. This is 2 features directly just from the population genetics data itself. First feature per gene was, what is the observed protein coding variation for the gene? And this is an empirical estimate of the underlying mutability of that gene. Next, we asked of those variants, how many of them were non-synonymous and also permitted to become common variants in the general population. So with just these 2 features, we essentially regressed the common functional variants on the total observed mutations in the gene, and we took the studentized residual of that as our RVIS, residual variation intolerance score. And what this does is it actually, by studentizing, it allows us to accommodate for the differing mutability of different genes. And essentially, this is the plot. Each plot dot here represents one of our 18,000 protein coding genes. The regression line is your indication of the genome-wide average for genes of similar mutability. As you go north of the line, what you find are genes where you see much more common non-synonymous variants than you would expect based on genes that have similar mutation rate. And as you go south, you see genes that have much less. And so this allowed us to quantify per gene its relative intolerance to functional variation relative to the rest of the genomes. And we can convert that into a percentile to just help us understand where along the spectrum of intolerance a gene actually resides. So this was all well, but we wanted to actually see if we could assess this and see how well this could be, how useful this could be in human genetics. So we took a number of gene lists, human disease gene lists, and for anybody interested, I've put the citation in where we've scrutinized quite a number of these. I'm just going to illustrate one as an example here. These are genes classified as disease-associated biome based on haploinsufficiency. And so what we find is if we take the 4% most intolerant genes in our genome, which is equivalent to about the 800 most intolerant genes that we have, they end up accounting for about 25% of all the known OMIM haploinsufficiency genes. And just to emphasize that, based on the construction of this score, all these gene lists and OMIM haploinsufficiency and all the others are actually orthogonal to the construction, so they don't actually influence the score in any way. And if you take the 25% most intolerant genes, you're explaining around a half of all haploinsufficient known human disease genes. So what I've described now is just our recent ability to understand or to quantify how intolerant a gene is. This is kind of your contextual information for a gene. And what I described earlier is the variant level scores that are available for how likely a given variant in a gene is to probabilistically damage that particular protein. And so you can immediately see the opportunity we have now to leverage both these pieces of information and try to improve the specificity of each by asking how often do we see variants that are highly predicted to be damaging to the protein happening specifically in proteins that don't like being played around with. And so to do this, we took de novo mutations. So these are mutations that have arisen de novo in individuals that have been ascertained for no clinical phenotype. So these are about 750 controls. Each dot here represents the de novo mutations found in this sampling. And we asked how many happen in this region here. So this is what we refer to as a hot zone. It's essentially a polyphen score greater than 0.95 happening in a gene that's among our 25% most intolerant genes. And the rate at which we see these de novo mutations in the hot zone is about 12% in controls. And so you could imagine that the obvious next question is how often does this happen in cases that have been ascertained for specific disorders or even for heterogeneous collection of disorders like in our sequencing clinics. So we've done this on a number of diseases. I'm just going to show our sequencing clinic results. So we looked at about 103 trios that were ascertained for heterogeneous collection of phenotypes, and we found that we see this strong preferential enrichment of the de novo mutations happening in the bottom right-hand corner where the hot zone is. It actually is about 41% of the known mutations found in this ascertainment happened in this hot zone region. And that quantifies to about 70% more mutations in this region than what we expect based on control trio data. And this can be translated to percentage population. And so if you think about it from the 750 trios that were sequenced, this quantifies to about 6% of individuals will have a hot zone de novo mutation. But then we have an additional piece of information which is from the animal studies, and these are essential genes. So we ask the additional information of how many of these hot zone de novo mutations also happen to occur in a gene that's known to cause lethality in mice. We've learned a lot from animal models, in particular from mouse models, and so genes when disrupted, if they're incompatible with life in mice, they're probably a set of genes enriched for some relevant developmental function in humans as well. And so we see a 2% rate in controls, and we ask how often do we see this in the sequencing clinic cohort. And you can see the enrichment is quite remarkably more significant. In actual fact, this is probably the strongest bioinformatics signature that we currently have to help us identify and prioritize putatively pathogenic variants in this case ascertainment. What we see is about 20% of these individuals compared to 2% in controls have a hot zone de novo mutation in an essential gene. That's a p-value of 10 to the negative 10, And that translates to about 90% more hot zone de novo mutations in the central genes that we see in this case collection. This could, almost translate to the fact that about 9 out of 10 times that we see a trio that comes through these clinics with a hot zone de novo mutation in one of these essential genes, it's probably because that trio has been, that de novo mutation is found because that trio has been ascertained for the phenotype. And so looking at the roughly 150 sequencing clinic trios that we've gone through, of these 20 hot zone de novo mutations in the essential genes, about 4 of these were actually triaged by this bioinformatics signature before they were securely implicated in human disease. And that's just on a set of about 100 or so trios. So clearly it's a great indication. It does not indicate pathogenicity, but it's an indication that you have an exceptional genotype happening in one of these children, and it should be carefully considered. So I'm going to describe 2 other bioinformatics signatures that we have that we find very helpful. We have quite a collection of these, but in this seminar I'm just going to describe 3 of them. So the next one is specifically looking for loss of function variants that happen in genes that are specifically intolerant to loss of function variation. So loss of function deficient genes. And the way we do this is, again, relying on a sampling of the human population. We take the 6,500 individuals from the exome sequencing project. For each gene, we have the underlying sequence context. We know what each possible mutation will result in. And using that information plus the underlying mutability of the gene, we can estimate how many loss of function variants we expect to see in a gene given how many variants they have, how many variants are observed in the gene. And so you'll find this way genes that have much less loss of function, unique loss of function variants than you would expect. And this is what we refer to as loss of function deficient genes. If you take a collection of controls, control trios again, and find their loss of function de novo mutations, this is important to emphasize. So loss of function de novo mutations do happen in controls as well. But what's important here is where they happen. So about 7% of the time, these loss of function de novo mutations are happening in one of these loss of function deficient genes. In comparison, when we look at the case collection, it's about 37% of the time. So we're finding loss of function de novo mutations in our sequencing clinic cohort about 80% more often than we would expect given what the control frequency is of these mutations. And finally, so the 2 that I've just described are bioinformatics signatures that really help us predominantly for dominant genetic models. And the one that I want to describe now is one of our recessive genotypes, and this is one of the more obvious ones, and that is identifying exceptional nullizygous genotypes. And so the way we can do this is yet again, take the sampling of the human population, and we count how many loss of function alleles are present in a gene. And we collapse the gene, and we collapse all those loss of function alleles in a gene to ask what is the frequency of loss of function alleles in the gene as a single unit. And then we can use that information to, and pretty traditional genetics formulas to ask what is the expected recessive rate of identifying an individual that will inherit 2 of these alleles. So your classic Q squared. And so by doing this, we can then convert that to how many non-consanguineous conceptions we would have to observe before we would see by chance an individual inherit 2 loss of function alleles, 1 from each parent. And then we set a threshold for sequencing clinic at the moment at about, we would require at least over 500,000 conceptions before we would expect to see 2 loss of function alleles land in that one individual for that gene. And so when we apply this to the sequencing collection plus some trios from our immunodeficiency collection, and we ask, of all the homozygous qualifying variants, how many fit this bioinformatics signature, we find that 6 of them do. And in fact, all 6 of those are actually genetic diagnoses. The 2 stars indicate genes where we've actually, as a group, been involved either primarily or secondarily in the identification of that gene with the association of that gene with disease. And by construction, this approach, this bioinformatics signature, is also robust to compound hets. And so we ask again, how many of the compound het genotypes, how many times do we see this? And it was 5 times, and of those 5, all of them were actually genetic diagnoses as well. So you see specificity here is pretty impressive based on just these 150 samplings of sequencing clinic cohort. And so what I've described up until now really is the work that we've been involved in trying to identify based on patterns of variation in the human genome, bioinformatics signatures that help us pinpoint things that are exceptional, that happen outside of what we would generally expect given what we know. What I want to talk about now is kind of how that actually translates to an N of 1 case. And I'm going to use the case of Bertrand Knight as an N of 1 example because it's one of our most impressive ones that we have. So we have full permission from the Knight family to present the photo and to discuss their story. Actually, the Knights have a blog up if anybody's interested to follow their diagnostic odyssey. Bertrand came to us at about 4 years of age of severe intellectual disability, altered LFTs, seizures, absenteeism, and involuntary movements. At the time, a disorder of glycosylation was I believe, considered, but the panel was done on known human glycosylation genes and was negative, and hence exome sequencing was followed up. When the exome sequence was interpreted by Dr. Anna Need and Dr. David Goldstein back then, what they identified was that Bertrand had inherited a frameshift variant from his healthy mother and a frameshift and a nonsense variant from his healthy father in a gene called NGLY1. Now, both mum and dad were clinically normal. They had no particular phenotype that would make them look suspicious, but Bertrand was fairly severely devastated. His phenotype was fairly severely devastating. NGLY1 itself is considered an intolerant gene, but more importantly, in the context of recessive genotypes, when we look at, given how many loss of function alleles are floating around in the general population in NGLY1, the expected rate of seeing an individual have 2 of these is about 1 in 10.5 million conceptions. And so this was not an indication that this was pathogenic, but it was a clear indication that something's happened here that doesn't usually happen and that we should probably consider this quite carefully. When the protein levels were assessed in mum, dad, and Bertrand, they were found to be decreased in the parents and virtually absent in Bertrand. Fortunately, his younger sister, who's healthy, had inherited neither of the variants, so she was not even a heterozygous carrier of one of those. But also, a lot of work was looked into NGLY1 and the actual biology of NGLY1. This was actually particularly important because when this was done, it was made pretty clear that NGLY1 could be an important candidate for this biology, for the pathway that one would expect to be involved in disorders of glycosylation. And so based on these 2 pieces of important information, it was actually considered by the genetics and the clinical team that the parents were able enough to understand if this candidate was communicated with them. And so it's communicated in a way that it was made clear that this is not a genetic diagnosis, but that this is a particularly interesting genotype. And we would have to wait for additional families before we could actually consider this a genetic diagnosis. And the families received this very well. Matt Might, who's actually has a PhD in computer science, created a blog about his son's odyssey and emphasized NGLY1. And this is also where the story gets a little bit interesting because in the next 3 or 4 years after this N of 1 discovery was made in NGLY1, I believe it's for every 1 case that was identified by the researchers that were involved, about 3 or 4 cases were identified by the mites through their blog. So families Googling, going through the same diagnostic odyssey and identifying that their children had, contacting the mites and then through the mites contacting researchers. And as a result of that, as a result of this, family communications, this actually now is the first identified deglycosylation syndrome, and NGLY1 is a securely implicated gene through double loss of function, complete loss of function. So what I've described up until now are ways that we can use patterns of genetic variation in the human population to understand things that are atypical and to better prioritize candidate genotypes. But we can also use the variation in the human population to help us clarify some possible misinterpretations. And these generally happen in genes that have already been implicated in disease. Some of the most important things to look out for, and one of them, the most important thing, in my opinion at least, is to at least look at the background genetic ancestry, a sampling of the background genetic ancestry for a trio before making too many conclusions on that variant. And I want to discuss this in a couple minutes here. There'll be a lot of information on this slide. But essentially, what we have here is 6,000 samples that have been sequenced by the IGM for various control or case ascertainments. And what's pretty obvious here is that there's an overrepresentation here from the European genetic background. So about 85% of these samples are from European ancestry. And so then the question is, if we ask for each one of these 6,000 samples, how many unique, so singleton, non-synonymous variants did they have in one of these OMIM disease-associated genes, and look at the distribution between people of European and non-European ancestry. This is the distribution that we get. So the blue distribution are individuals that have European genetic background, and red is a combination of non-European populations. And what you find, the way one would interpret this, is that for any given proband that comes in of European ancestry, we would find about 15 unique non-synonymous missense variants in one of these disease-associated genes, compared to about 51 if that individual came from a South Asian background. And so having more candidate variants in OMIM disease-associated genes does not translate to being more likely to find the pathogenic variant. It's actually the opposite problem. You're more likely to create a narrative around some variant which seems to look interesting because it happens to land in a gene that could be have some narrative with the patient ascertainment, etc. And so we wanted to see what the effect would be if we added an additional 60,000 samples to the reference cohort, but particularly adding additional representation from the ethnic minorities. And so we took the exact collection. This is the exact consortia. And what we see here now is that while Europeans are still the majority representation, each one of the minorities actually get a lot more, are starting to take up a lot more of the pie. And so the effect this has is an expected effect. We see a harmonization of the two distributions. Now, if we use this reference cohort of 67,000 individuals to find unique variants, we find about 6.5 per European proband, compared to about 12.7 per South Asian proband. And so this is still a twofold difference. Twofold might not sound a lot, but it's actually quite a lot when you consider each one of these patients that come through the door. And so what I want to go through now is an actual observation of how this actually translates to becoming a bit of an issue. This is a real-world example where we had a colleague of ours from Duke, a pediatric neurologist, contact us about being contacted by a family where the sequencing diagnostic company had identified a likely pathogenic KCNT1 variant in the child. Now this was important because the child had severe infantile epilepsy, which can be linked to a KCNT1-like epilepsy. The variant itself was not present in that population reference panel that was used. However, at the time, father was unavailable, so mother did not have this variant, child did. It was not seen in the reference panel that they had. And what's important here is that if this variant was actually pathogenic and resulted in a gain of function like most of the KCNT1 positive epilepsies do, then there could have been an opportunity for trialing a FDA-approved compound called quinidine that has been shown in oocyte models to actually bring down the gain of function effect in the actual mutations that patients have had. And so this became important, and Dr. McCarty approached us and asked us what we thought, and so we asked for each of the ethnic strata that we have, what does it look like in each of the strata. And what we have here is a principal components plot, so this is an ethnicity plot. The two axes are two ancestry axes. This group here is the European group, and the only two genetic backgrounds we've introduced in this plot are Europeans and Middle Eastern, individuals of Middle Eastern background. But we then have self-declared ethnicity on some of these, and so we wanted to ask how many of these individuals are self-declared Ashkenazi Jewish background, and we see that it's quite clear that this bottom cluster among the Middle Eastern group is representing the Ashkenazi Jewish population, and these individuals here are likely to be individuals that have a grandparent of Ashkenazi Jewish descent, so they're along the admixture spectrum. We then asked, do we see this variant anywhere in this population? And it turns out when you ask how many of these samples have the variant, we find that about close to 1.5% of the Ashkenazi Jewish population that we have in-house has this variant present. So on that basis, it's very unlikely that this is a pathogenic variant. While we did not communicate back to the family, it's very likely that the father who's unavailable is of Ashkenazi Jewish descent. And you can see here that this variance, at least within our European cohort, also never observed. So I'm going to stop there on the N of 1 and now move across onto how we can actually, onto the second paradigm, which was the case control comparison. And one of the ways that we do these analyses is by collapsing analyses, where we now ask for a genomic region, so not rather than a specific variant, for a genomic region, do we see more qualifying variants in a case or control population than the other? And so we can define qualifying variants using certain criteria, the most frequent one being population frequency. So if you're looking at very rare disorder, severe, and you expect it to be highly penetrant model, then you would probably have a basement level monoallele frequency that you would permit, and also what kind of effect that variant is predicted to have. And then you ask, is that region, most often a protein coding gene, significantly enriched in one population versus the other for these qualifying variants? And this is an important actual framework because it has a number of relative strengths over the N of 1. One being that it has high power, especially when the expected effect of the variance is to be a large effect. And recruitment is often easier than when you have to recruit a pedigree or parents, especially if it's an adult onset disorder, something like ALS, which I'll describe some results from, where by the time the disease has presented, parents are usually unavailable. Some of the weaknesses are not unique to case control, to collapsing analysis. These are issues that we've been dealing with in statistical genetics for a very long time. So comparability issues between the technologies, so differences in sequencing characteristics between your cases and controls can result in cryptic bias that creeps into your results. Population stratification is one that's been well understood. But through the pipeline that we've developed, we've actually spent quite a lot of time trying to understand what are the possible sources of bias that could influence results, how can we mediate these, and actually then delivering approaches to actually accommodate for these. And I'm not going to go into all of them. A couple here that I've highlighted are things like opportunity-based bias, and this one's quite important because when you exome sequence an individual, you can get variable coverage for different parts of the protein coding exome. And if you tend to have your cases sequenced at a given time that's different to controls, it could be that that kit used at that time covered an exome better. And so what we do is we actually harmonize the real estate that we run the test on to make sure that both cases and controls had equal opportunity to call variants in them, and we discard exons where there's high variability in calling properties between case and controls. And that usually ends up being only about 6 to 7% of the protein coding exome that we don't interpret because of this variability issue. And also ethnic inequality, we can accommodate for that by actually getting ethnic predictions, so eigenvectors and principal components, from the exome sequence data for individuals. And so if you don't do any correction for these tests, and this is the result, a QQ plot based on a case control permuted analysis, so there's actually no disease information here. What you see is, so for those of you that, I assume many of you will be familiar, but you have your expected p-values here based on the number of tests that are being performed and your observed p-values. And what we see here in this illustration is a statistical geneticist's worst nightmare. You see clear genomic inflation throughout the genome. And in fact, if this were from a disease study and you did not correct for anything, you would identify 60 genes that are genome-wide significant, which clearly they are not. So we accommodate for these using a number of adaptations to the framework. And so this is what it looks like then when you actually have accommodated for the potential sources of bias. This is a QQ plot from a study done by Liz Cerulli from our group. And this was based on about 3,000 ALS cases and about 6,500 controls. And what we see here is that out of the 18,000 protein coding genes that were assessed from a collapsing analysis, we see 2 out of about a dozen to 20 well-known ALS genes, the top ranked in the entire genome, one of which is already genome-wide significant. And so what this illustrates is, just based on the ranking, the likelihood of seeing these real ALS genes up there in the top ranked genes is pretty unlikely to happen by chance. And what that could indicate is that within this set of genes that are slightly elevated, with more power, you will start to see more of these actually become genome-wide significant. So what we did is we took a subset of these that were elevating, and we sequenced them in additional cases and controls. And one gene in particular, TBK1, we found that in a replication cohort was significant at 6 to the negative 7. And most importantly, when you take the combined evidence for this gene, we found genome-wide significant, clear, comfortable genome-wide significance at about 4 to negative 11. And if you consider how often these qualifying variants happen in controls, it quantifies to about 1% of these sporadic ALS cases have one of these TBK1 variants, either because it's contributing or causal to their ascertainment for ALS. We've also looked at this in a number of other diseases. I'm just going to present one more recent one, which is a public paper that's currently in preparation. This is based on non-acquired focal epilepsies with a family history. So here we only have 600 samples, and we have about 3,500 controls. And the qualifying variant criteria here are looking specifically for variants that are predicted to be loss of function or probably pathogenic by polyphen, and also variants that are singletons, including the exact reference cohort. So this allows us to get down to a frequency below around 0.001%. So we're getting pretty far down the frequency spectrum. And what we identify is, when we look at this cohort, we see really nice liftoff here for a number of genes. In actual fact, the top 4 genes out of the 18,000 genes genome-wide are well-known bona fide epilepsy genes. The top 2, which are actually genome-wide significant here, DEPDEC5 and LGR1, which are both well-known epilepsy genes. This cohort wasn't ascertained on the basis of knowing they had these mutations, so that's an important point. And the probability of finding your top 4 genes genome-wide being known dominant epilepsy genes is somewhere around the lines of 10 to the negative 11. So it's highly improbable that we would see 4 of about 35 dominant epilepsy genes ranked top 4 by chance. And so this has a number of interesting interpretations. One is that the focal epilepsies, based on these results at least, appear like they might have lower locus heterogeneity than we might have originally anticipated, at least some of us in the community. Based on this collection of 586 samples, about 8.5% of these cases have a qualifying variant in one of these 5 epilepsy genes, and the expected rate based on how many we observe in controls is only about 0.7%. And most importantly is that this data suggests that this framework, the way that we're analysing these data, is very likely to help us identify additional genes and securely implicate new focal epilepsy genes with additional case collections. So in the last 2 slides, I just want to talk briefly about some of the work that we've done in non-coding. So it's going to completely shift from the first 2 topics. As I mentioned, most of the work that's been focused on in human genetics has been on the protein coding exome, which is about 1.5% of our entire genome. What we wanted to start and try and do is see whether the patterns of genetic standing variation in the human population in the non-coding sequence could be interesting and informative in the context of human disease. And what we did is we took each gene's immediate non-coding sequence, so 5' and 3' UTRs, and 250 base pairs upstream of the gene, and we assessed its underlying mutability based on the mutation rate using a trimer matrix. And we took Y to be, because we don't have an indicator in the non-coding region of functionality, or a good indicator of functionality and non-functionality, we take it as just all common variants that happen in the non-coding sequence for that gene. And this is based on about a sampling of 700 whole genome sequence samples. And like the original ARBUS, we create an NC ARBUS, and this is the regression plot that you see. And so it's interpreted in a similar way. The genes that are northwards of the regression line are genes where we see much more common variation in the non-coding sequence than we expect, based on genes whose non-coding sequence has equivalent mutability, and genes south are those where we see much less. And so an important, one hypothesis that we had here was, it's well known that the non-coding sequence, especially the 5' and 3' UTRs, are pretty important in gene dosage, regulation of genes, in mRNA stability, etc. And so one hypothesis is that the non-coding sequence could be, or patterns in that non-coding sequence could be correlated or associated with genes that don't tolerate loss of function variation, being haploinsufficiency or triplo sensitivity. And so we went ahead and took that list of loss of function deficient genes, which were based on the 65,000 exomes, and we looked at whether those genes preferentially have more intolerant non-coding sequence. And this is a violin plot showing that it is true that based on these data, the non-coding sequence of loss of function deficient genes has significantly more intolerant non-coding regulatory sequence than that of loss of function control genes, which were defined as genes where we see about as much or more loss of function variance than we expect. We also looked at human haploinsufficient genes based on ClinGen's gene dosage sensitivity map. It's a highly curated list of genes. And again, we see that the non-coding intolerance, so the patterns of standing variation and non-coding sequence of genes, is highly predictive of genes that are haploinsufficient, that cause disease by haploinsufficiency in humans. And that's at about 10 to the negative 15 with an AUC of about 0.66 there. So what I wanted to emphasize here is that it's pretty clear from a lot of the work that's been done in the community, especially in the past few years, that next generation sequencing has revolutionized the way that we do medical genetics, particularly how we interpret individual patient genomes. By understanding the typical patterns of background variation in large human populations, we can now understand and identify bioinformatic signatures to help us in interpreting individual genomes. And this has only been happening, this work's only been made possible in the last few years since these exome sequence data have been available. And I do feel like we're just scratching the surface, and a lot more is going to come out of this space in the years to come.